Good morning and namaskar. First of all, I'd like to convey my sincere thanks and gratitude to Professor N. Udupa, who has been instrumental to organize this webinar, particularly on Covid-19, which is, I believe, is a very timely topic. He's the president of IAPST, and he's very much, the man who can do lots of things, and who has been doing lots of things for the organization. We are all proud of him. Now, I also like to convey my sincere gratitude and thanks to all the other speakers who already have delivered or will deliver some lectures today. And of course, the other persons who are the organizer or who are the helping hands of Professor N. Udupa, and of course, the persons who are participating. Now, today, my topic is COVID-19. Where are we? This COVID-19 is very, very interesting virus. If I just look at it, or if we like to explain it, I preferably want to restrict this lecture to in a very simple and common way, but with some science also. Basically, if we look at this virus, this is an RNA virus. So RNA virus has some problems. Most of the cases, RNA virus, RNA polymerase is basically, it has no editing, devices. So naturally, normally RNA viruses are very, very susceptible for mutation. So this is number one problem. Secondly, this virus is quite unique in its designing. Whether it is naturally or in a synthetic manner, it is hard for me to tell you. But this particular virus genome has basically two parts. One part is normally the common corona or adenoviral type of genomic sequences, where basically they like to infect the people very quickly. And the other part is the retrovirus gene. That means, kind of HIV virus gene. So that part basically is they have the capability to mutate and to hide in your system. And of course, it can remain in your system even for a longer period. Normally, these viruses, so basically two types of part is there. One, it will infect you first, quickly, I want to mean. And then the second part is, when you are infected, what will happen? Basically, it will remain in your body for a longer time. This is a new virus. So much, knowledge is not there with us that how long it will remain in the system. Many results, many findings, many literatures are giving different types of answers. I have found that they are actually giving answers for 14 days sometimes 30 days, sometimes in 48 days. What is the reason? Reason is, I said, like HIV, they can, at the early stage of infection, they can hide in the macrophages. They can hide in the cells which actually present, the antigen. So antigen presenting cells. So that is a dangerous thing. And because of that, sometimes you may not get the virus in your saliva or in your, throat samples, but basically it may remain in your, like antigen presenting cells, or these sometimes, like as I said, dendritic cells, macrophages, or this type of cells. So this is really a matter to think over. And in the slide, if you just look at this slide, basically it is actually of SARS, originating from China first in 2002, and then you see we have lots of similar infection we have found in Middle East also, in terms of the, its nomenclature, coronavirus also, but it's called MERS. And because of some maybe alteration of this genomic sequence, and of course the present one, COVID-19. So basically what it is doing, it is actually causing some infection like pneumonia, like respiratory failure, septic shock, death. But before going to the other detail, I just like to say, is it causing this thing to all? No, this is not. That means if you are infected, it is not that you may have all these type of symptoms. Sometimes even diarrhea is there, confusion is there, some abnormalities in your movement is there, little, or I can say you can have some kind of problem to speak properly. It is also there, reported. So lots of similar symptoms are there, but it is not killing everybody. It is just, you can say 80 to 85% people, those who are having this infection, the symptoms are not coming. Even if it is coming, it's coming in a very, very less degree, and they even sometimes they don't know that they're suffering from COVID, and they get rid of it maybe in the normal course of their life. But the infection, when it is coming to the people, there's 15% of the people, what happened, they sometimes develop these symptoms. And out of this, the majority, this 15% of these people, when they're developing symptoms, most of the cases, they're having this pneumonia type problem, like, the breathing, respiratory problem, and then ultimately, even sometimes, some cases, report is there, it is causing some hemorrhages in the cells of heart, cells of other, even lungs. So, how it is working and how this virus is actually causing problem to those people, why not it is causing the problem to others? Is it because of those people are actually developing the immunity or developing the antibody quite late? So all this investigation is needed to find out what is happening. But the major complication, whatever I have said, the major complication is being, it is coming up simply because of the two factors. One is, as it is claimed in the literature, cytokine storm. So all on a sudden, the cytokines are coming up in your system at a very, very high level compared to the normal. The question is why? Or how and why it is coming may not be known till. There are lots of, researches going on. People are detecting, people are speculating, people are thinking that this could be that, this could be this. Fine. But that cytokine storms are actually the killer of your cells, killer of your tissue, damages your organ. And that is actually the ultimate cause of, or that is actually leading to the death of the people. Now, what is the problem, this cytokine storm, if it is there? I said what is cytokine storm, I think you have understood. There is the high level of cytokines, this enzyme is coming out, and that is actually, normally it is required to fight yourself against the, foreign bodies, bacteria, even viruses. I do not know how, even sometimes for certain other toxic materials also. But you see, the major problem of this is when it is coming up at a very high level, it is working against you. So what does it mean? It means not only virus or viruses are damaging your system, but also the cytokines are damaging your system. So normally we say in case of medicine, synergistic effect, but that is for the sake of benefit. But it is the similar effect, combined effect, not for the sake of benefit. It is for the sake of further damaging of your system. So it will damage your tissues, organs, and that causes the multi-organ failure, and the patient will die. So if we look at this virus, although the picture is quite colorful picture, basically interesting part is that normally all viruses has a protein capsid, that is the cover cell, and inside it the nucleic acid is there, either DNA or RNA. I said it is RNA in this particular case, and the protein coverage capsid is there. But along with that, of course, there are some different types of proteins normally there. Sometimes some proteins actually there, some part of the genome, some part of the nucleic acid actually develop its core. Some parts works for different other purposes to give the shape of this virus. That I will come maybe later on. But at this stage, if we just look at this slide, not only just simply the protein part is there, and of course there are some projected parts. There are some prolongated part that is also protein, glycoprotein, and what you call this is basically quite interesting, because they help the virus to bind with the cells with some receptors. So, and of course it has HE, that is hemagglutinin esterase protein, so that is also playing some role. So you see, this virus structure is not very abnormal. It is not very uncommon. So you can say it's a common structure of the virus, and yes, but it functions in a different way. Now if we just go for the different types of COVIDs, if we just go for the different coronaviruses, you see, you will get some differences in the genomic sequences. Maybe it will not be very interesting for many of you, but anyway, some differences are mentioned here with beta, alpha, gamma, and delta type of COVID viruses, and what are the special thing there, what is not there, all these things are mentioned in this particular type of structure. Basically, this could be important. Those who are interested to understand is sequence. Now, if we go for the genomic structure in a very simple way, if we want to describe, as I say, that it has H gene, and along with that, of course, this special prolongated protein, which is commonly called spike protein, and this has unique N-terminal fragment. So this part is very important, and of course, like it has ORF is, basically it's encoding 9680, nearly 10,000 amino acid polyproteins. So this is also very interesting. Then normally this is not very uncommon, but this is very interesting. It has this ORF 67% of the genome, 67%, which encodes 16 non-structural protein. I said a little early that in case of the virus, there are two types of many important protein its genome synthesizes. One is structural type of protein. This is basically needed to develop its capsid, but the other case, the other part is non-structural protein, and non-structural protein, they have some roles. They actually have some roles for the development of its nucleic acid sometimes to play role in its, invading the virus or doing some help to the virus for its survival, for its proliferation, or what you call this for its new generation, etc. So the structural and non-structural proteins are there, and this non-structural protein, and another I said that this prolongated protein, glycoprotein, S-glycoprotein, basically this helps as the receptor binding domain. Little earlier I said that thing. Now this slide I want to just quit because this is basically the matching and differences have been shown in different types of SARS, MERS and COVID viruses, coronaviruses. Now here also this is basically the comparison of this genomic sequences. What are the differences? In the earlier picture, this is just to kind of projection, pictorial projection, but here it is just briefly described that what are the amino acids, what are the amino acids, and of course the substitute, what are the differences basically, etc. But this differences is of course virus to virus, even human to human, human to animal. So these differences are always there. But the major thing is not these differences. We have to understand, we have to find out how or what role these differences are actually playing. This is important to understand. This part is it is just shown this which part of this genomic sequence produces the non-structural protein and even the spike protein, rich by etc. Structural protein, non-structural protein, all these things that have been described. And these non-structural polyprotein numbers, it is also mentioned here, so you can easily understand or differentiate different types of COVID. Now, if we just look at this coronavirus, COVID-19, how this coronavirus is normally enter into the cells and then infect. Infect means, they're infecting us, but they are actually doing this thing for their survival. They want to proliferate. They want to generate new virus. So on the cell receptor, this particular projected protein, what you call this spike protein, little earlier I said, so they bind with the receptor. They bind with the receptor. You can see it here, just here. These are the receptor, cell surface receptor. They're binding with this receptor, and then they enter. Sometimes they enter by endocytosis also. So once they enter, then like other viruses, they normally just try to their normal function. Their genome normally comes out of the virus, and then they take the control of the cells, and then they develop their own proteins, they develop their own nucleic acids, and then the packaging and the new viruses comes out. So it could be the ultimate thing, virus release. This projected virus actually, the projected proteins bind with this receptor, and then they come, and then actually they proliferate, and then they go out, but go out of the cells and infect the new cells. So thereby, number increases, we say viral load, and if the viral load enhances, obviously your cells, tissues will damage. The body will try to protect. They will release different types of proteins. They will release cytokines, and then abrupt, high level of release of cytokines actually start destruction of your own tissue, own damage, causing the total system jeopardized, rather I should say kneel down to your survival, kneel down to your immunity. So actually this is a dangerous thing, thereby it is killing that those number of people. What I have found, on an average, it kills about, say, 5 to 7% of the people. Sometimes in some country, percentage of people is less, sometimes it is more. But if you just look at the picture in a different way, out of 100, 85% people, they are not actually having any problem. So the people facing problem is just 15%. And out of this 15%, if 5 people dies, that means if people who are being hospitalized, if the number is 15 out of 100, out of that 5 are dying. That means they will pay the debt of the nature. And the thing is that causes, 5 out of 15, that means nearly 33%, one-third of the hospitalized patients have the possibility to die. Now, human coronavirus infects different types of cells. What are the cells it can affect, it can infect, so it is there, and you can see, this is just, I like to say that this is just the initial knowledge, preliminary knowledge. I don't agree with that basically, because you see, in case of this COVID, as per the report, initially it was actually infecting respiratory epithelial cells and T cells. T cells, of course, because main fighting will be given by T cell. Particularly, I like to say CD8 plus and CD4 plus. Anyway, so these cells basically are involved, but simultaneously also the other fighting cells from lymph node and of course the sources, tissue areas like spleen, etc. But nowadays, with the time, lots of changes are there. We found that initially it was believed that it doesn't infect the intestinal cell, it doesn't infect the other cells, other tissues, but it is not true. It is infecting other tissues also. With the days, with the different kinds of literature, with the different kinds of publication, we came to know, we are just coming to know that yes, this virus can infect other organs too. Even some people are suffering from diarrhea and no other symptom is there, and when they are tested, it is found that the COVID positive. So, COVID-19 positive. So, this is something that lots of things are yet to come, lots of things we should know and we will know. This may be a little bit complicated picture. Infection process of COVID-19. Infection process, basically, the same thing, whatever I have said, but here what is mentioned is basically the different proteins involved, what are the proteins, and that projected function of them. But basically, even I like to say that this is also in the initial stage. Lots of things are unknown yet. We have to know exactly how this virus causing infection, how this virus, or what are the proteins, what basically, other than this normal common protein which I mentioned, what are the new proteins, what are the new areas through which it is infecting, it is killing, it is infecting the cell, it is proliferating in the cells. So these things should be known, and this is important to develop the medicine against it. Now, possible way to combat this is, I do not like to say much about that, because every country, everywhere, government or different media, national media, they are actually giving lots of information to all of us. how to protect yourself. Of course, few things are important, that you have to use mask. Mask is very important. You have to make a safe distance for virus, normal viruses, or we say, which a very infecting virus which can cause quick infection. The safe distance with the mask, if you use mask and other protection, safe distance, you can say, normally people believe 8 feet, but for this you can take another 2 feet or maybe 4 feet, that is 10 to 12 feet distance. You should always maintain from the unknown people or the people whom you do not know whether he is suffering from or not. So, for those people, for unknown people, where you have the doubt that I do not know about the person is having the asymptomatic viral infection, or we can say the asymptomatic people and also the symptomatic, you should maintain at least a distance of 12 feet, 10 to 12 feet. Then use your mask, and of course you have to wash your hand, every 1 hour or whenever you are touching some unknown thing. After that, it is better to use the soap. Soap, I want to mean the lots of liquid soap in India. We know lots of good companies are there. So you use this liquid soap and wash it. That is the first choice. But sometimes, you may not have that chance, opportunity to get water, get liquid soap and all these things to clean your hand. It is not possible sometimes. When you are traveling, when you are on the journey or somewhere else. So always keep hand sanitizer of good company. It is very important that should have 70% isopropyl alcohol or ethanol. This is very important. Ethanol, normally you may not get it, but with the isopropyl alcohol. So the WHO has already, they have mentioned that what are the chemicals basically required for developing this hand sanitizer. It is there. Anybody can go and check from the WHO side. Even they can prepare it in their house. You don't need, isopropyl alcohol is also not that costly. It is not a very costly chemical. So you can prepare it for yourself in the house. And of course, it is better for just to keep yourself safe from the unknown people. Never try to avoid the crowd, try to avoid the crowded area, crowded, market, even the shopping mall or whatever, So it is important to keep yourself safe at a distance from the people. Sometimes, of course, you have to, whenever you are going out, you have to use mask, and of course, the hand sanitizer should be there, and that is the best way. Try to avoid any touch with your hands or with anything to your face. Till you come back to your house, wash your hand, clean your hands, or you should come back to the place where you can clean your hand, and then clean your hand, of course, with soap for at least 20 seconds, and then you can do it. Now the medicine. This is very important. What medicine we should take? What are the medicine available? The question is, at this moment, really there is no medicine available. It is already there, lots of, kind of idea. With the idea, people are testing, and then the different way they have developed certain combinations, certain medicine. One is this hydroxychloroquine. This has been used in India, and of course other acyclovir group of drug, and these are just now currently available, like remdesivir and also favipiravir. So this they're all acyclovir type of drug. Basically, what I feel, it's a question of, it's just a testing procedure. Testing procedure, I said that really 80% of the people, they don't need medicine, 80 to 85%. They don't need anything. They will recover. What they need plenty of liquid. Plenty of liquid. Basically, for any viral infection, you have to consume plenty of liquid. Basically, liquid means not only water, you can take fruit juice, you can take milk, you can take, like even some other, you can say the semi-solid foods, or even some proteinous fruits, like, the proteinous food also, like basically fish, chicken, light proteinous food. So this type of things is important. Even proteinous fruit can be taken, those who are vegetarian type. But that should be semi-boiled. That should be boiled, that should be soft. That is semi-solid mass should be there. It should not be very hard. For the first at least 7-8 days of infection. And then you can take, along with the other food, easily digestible food, you can continue to take lots of, liquid food. Here, vitamin C is essential, because vitamin C is essential for any viral infection. Antioxidant property is there. So you can take lemon, you can take, the lots of, I can say the lemon juice and other fruits where vitamin C is there, like guava, etc. Along with that, another very important vitamin is there, vitamin D, and that is required. And for that, you should have regular exposure to sun. Maybe half an hour or 15-20 minutes, as much you feel comfortable with it. In the morning sun, you can just be there. So, yes, of course, as I said, these things are required. The quantity has been even mentioned, but of course, I believe that micronutrients are essential for the body to fight against any infection, any problem, any pathological damages also. So micronutrients, they have enormous function, including this zinc and also, the vitamins. So as it is advisable, they should be taken, that they could be even, prophylactic. That means you can take even regularly to keep yourself protected from this viral infection. Again, I just like to say some experimental treatment strategies, whatever I have said. Why chloroquine? Why hydroxychloroquine? Why this, different other antiviral drugs? What are their function? How they actually damaging the virus? That is mentioned here. You can understand that what are their role, how they are damaging. But here I like to say that it has not been tested actually, but it should be tested also, even first in vitro, and then people can use it in human being also, particularly when the condition is serious. Why not we should test, since the virus has pseudo-retroviral gene, why not we are testing, why not we are giving this acyclovir group of drug, along with that some antiretroviral drug. Example is like didanosine, stabudin may not be so. Whatever you see, this type of drug could be chosen, could be tested, at least in vitro way or in some serious patient, it could be checked how these drugs can work on this virus, because simply it is not yet tested, but it should be tested. If the opportunities are there in some laboratory, they should conduct against this, the in vitro test against this virus, and then try to find out whether really this combination could be effective. You never know. You never know. It could be effective also, because it has the pseudoretroviral gene. Now, the standard test and recommended for this protection gown. Protection gown, we normally say it is PPE. You know, PPE doesn't mean only the gown, even the goggles, even the gloves, even the shoes, even the mask, even the head cover, all these things are coming under PPE. So in Indian scenario, I like to just highlight few things before conclusion. How much we are actually doing and how much we need. Actually, for this PPE, lots of, reports came from different, media, national media, newspaper, but they should have a standard. The standard, whatever I have mentioned here, the standard is recommended by US FDA. So why don't we follow? Why our organization, those who are preferring this, PPE, why they are not following this particular guideline? Like, the material should have proper tensile strength, they should have tear resistance, maybe it is there, but that resistance should not be much more, should not be much less also, within the range it should be there. And link generation, of course, water vapor resistance transmission, and this is very important, breathability and steam strain. So this thing, as per the guidance, they should be prepared. I am not saying that these are all enough guidance. Maybe certain other things which authority believes, regulatory authority believes that it is required to impose, that should be there. Why I am discussing this thing, I will tell you, because the present PPE is also not good enough. Why, I will discuss. Of course, the sterility for the, particularly for the gloves, sterility, freedom of, freedom from holes and the other dimension, etc. All these things should be checked as it is recommended by different pharmacopoeia. I think our pharmacopoeia should include this thing. And the dimension of the gloves, little earlier I was saying that these are the mentioned dimension. So, and of course the tolerance is equally important, that is there. Mask, some guideline is there about the mask, and what type of mask we should use, what are the standards, what is required, what is not required. The mask should have good breathability. This is very important. So, making the mask N95, it is good, but the breathability should be there. It should not be that once you are using or one patient is using, they suffer from breathing problem because of this pneumonia or lungs infection, and you are just giving him to put on and to wait to hospital and waiting there for 3-4 hours and all this thing. He may die because of the insufficiency of oxygen. So that thing we should understand. Of course, here there are different important points regarding shoes and face shield. Whatever I was mentioning, it is there. So it is available in the different guidelines, and accordingly we also should follow this so that we could be comfortable. Now this is, you can say, the unique example of this COVID-19 PPE. This PPE causing so much, unbearable heat. That is, this comfort is not there, discomfort that the person who are wearing. It is in case of this is a nursing staff. Person, those who are wearing, they are unable to put it on. So this is very dangerous because the patient, if he puts on for longer hours, or even the health staff, this causes discomfort, and that discomfort, this famous case of the Russian nurse who did not actually she left her clothing and just put it on. But there also you are not providing the safety. There also this PPE must be opaque, so that the person who are feeling discomfort, who are feeling not comfort, they should remove their clothing and put it on, and that person's body cannot be visible. So this is very important that PPE should be comfortable to wear, number one, and secondly, PPE should be such, it should be opaque, so that the body of the person should not be visible. Now, the PPE and all these, agent, all these things I have just mentioned, we have just recommended something here. We believe that in case of India, this should have universal standard. Of course, it should have, it should be opaque, and it should be such that it should be comfortable to put on. It should not be uncomfortable. All the countries in the world, all the researchers, particularly those who are working with this type of material, they should try to think of, they should try to develop, or lots of research really is required, to develop such materials which will be comfortable to put on. Because if I am uncomfortable, I cannot treat a person. If a patient feels uncomfortable with the clothing, he will not try to put it on, and that will deteriorate his mental health and physical health condition. So we should understand it. Now, this is interesting. This is the hand sanitizer. This is available everywhere, and lots of bad things are being done by selling this hand sanitizer, which are basically a wrong one. That means the composition is wrong. They are not giving this really 70% of this isopropanol or alcohol, ethanol. They are mixing some unwanted chemicals which are causing lots of irritation to your skin, damage, or maybe some kind of allergy or this thing. So lots of things should be cared. And I just like to show you this level of, disinfecting agent. What you can see, there is no manufacturing date, there is no expiry date, but this materials is being sold. So care should be taken to protect us, protect ourselves, and everybody, every people should just join their hand. Every organization should join their hand to develop this system which can really fight against this COVID. I can say that this COVID infection is really killing number of people, but it cannot kill 100% of the population. Really 85%, 80 to 85% populations are fighting and winning it. Remaining out of this 15, maybe 1/3 is dying and the remaining are surviving. But question is, why 1/3 people will die? What is their fault? What is their problem? They are innocent people. They never might have seen this virus, but these viruses are infecting them. So it is the time for all the health workers, it is the time for all the researchers to find out the real truth, to find out how to tackle, how to fight against this COVID virus, and that should not be not too late, because delaying will kill maybe lots of many people. So we have to be first, we should take special initiative, we should try to help each other to develop certain mechanism to fight against this COVID properly, not in a very scattered way, not just like in a very unknowing way. There should be a proper schedule, program for this, and how the common people should know exactly how I can protect myself, just simply using this thing and that. It is there, but simultaneously it is very important to maintain this distance. Till we get this vaccine, we have to find out this mechanism, join our hand together to fight against this. But let me tell you another doubt in my mind, that this vaccine may not cause a long-term immunity. Question is, if this vaccine can develop this memory cells, and can recognize this virus in future after the infection, or even once you are vaccinated, it can remain in the memory, only then it will be successful vaccine. Otherwise, its vaccination maybe every 3-4 months or 6 months you have to take. But whatever, till we get the better opportunity, we have to fight with this. The dangerous thing of this virus, it can hide in our system. So thank you very much. I wish all of you a successful evening, and you can learn lots of things from the learned speakers who will just deliver also, and those who have delivered. Thank you very much. Thank you all. Thank you, Professor Uduppa.